Here we are in the Ramsar listed mangrove wetland of Tower Point in Sydney, and I want to talk a little bit about sampling of autotrophs, that is, organisms that are capturing carbon dioxide from the atmosphere and converting into organic carbon. They are the level of the ecosystem that powers everything else that's going on in the wetland. If we want to know what the trophic dependencies of different organisms are, we really need to understand where the carbon's coming from. Which of all these different sources of autotrophic carbon are the most important? And then we can focus our management strategies on conserving those important primary sources of productivity. So we think about what is capturing carbon dioxide here. There's a number of candidates. One would be the mangrove trees themselves. And here we have Avicennia marina, which is one of the most commonly distributed mangroves in the world. And I can easily sample some fresh leaves of the Avicennia tree. We can prepare those, I can pop those in a sample bag, and we can prepare those for isotopic analysis. Looking around me, there's quite a bit of carbon that's being fixed in the leaf. However, there are some processes that occur in the mangrove that create subtle variations in isotopic signatures between components of the mangrove tree and also parts of the mangrove at different age. And here we have, just for one example, one of the leaves that has fallen from the tree and begun to decompose. Now that's a more likely candidate for fish or crabs to be eating. And the fact that it's undergoing decomposition also means that it might have a slightly different isotope signature to the fresh leaves. So here we already have two candidates. We have the fresh leaf and we also have the decomposing leaf. And what I'd want to do is wash both of those in the laboratory and then submit them for isotopic analysis. It might be that in the process of decomposition, the isotope signature of the dead leaf has changed. So there's two. Thirdly, we have the mangrove root system. And I'm just going to be sampling for the moment the above ground component of the root system, which is the pneumatophore. About 80% of the carbon, perhaps more in some cases, occurs below the ground. And so given that a lot of the primary consumers, a lot of the first level consumers in the mangrove forest are benthic organisms that are burrowing into the soil, perhaps the carbon that's in the soil is making an important contribution. So I want to be sampling that carbon, and one of the interesting things we've discovered in recent years is that the isotopic signatures of the roots can be quite different to the leaves. And so now already we have three different components of the autotrophic carbon which may have different signatures: the leaf, the fresh leaf, the decomposing leaf, and the root system. If we look at the pneumatophores themselves, you may notice that there's some algae growing on the pneumatophore, filamentous green algae, and that is an additional source of carbon that will have a different isotope signature. So there's another potential source of carbon that might be contributing to the food chains. Here we have some seagrass rack that's been washed into the mangrove. Now, seagrass has a very enriched carbon isotope signature compared to mangroves. And if this is contributing to the feeding behavior of crabs and fish in the wetland, it's going to be very obvious when we look at the carbon signatures. So if we can see this seagrass rack in the wetland, it's going to be a really interesting thing to get a signature on and look at the extent to which that source of carbon is contributing to the overall food chain. One final opportunity for organisms is to just feed on the surface algae that forms within the matrix of the mud right on the surface of the wetland. We call it microphytobenthos, unicellular algae that by biomass contributes enormously to this wetland. By sampling the very thin surface layer of the wetland, we have an additional source of carbon, the microphytobenthos, the surface organic material that might just be an important component of the diet of crabs. Often if you look at them feeding in these environments, it does look like they're just feeding on the mud. They're filtering out the microphytobenthos from that surface organic matter. So plenty of things to think about. You know, it's not just the mangrove leaves that we want to be looking at here. We want to be looking at old and new leaves. We want to be looking at the mangrove root systems. We want to be looking at what's on the surface of the marsh, the algae that's growing on the roots. We want to look at the seagrass rack, if that's coming into the wetland. We want to be looking at the microphytobenthos. Before we start our trophic investigations, we want to be thinking about all of these potential sources of carbon, how we might sample them in the wetland.